Okie dokie. Hello, everybody, and welcome. Thank you for joining us today. We are very pleased to be welcoming Dr. Travis Craddock, an associate professor in the Department of Biology at the University of Waterloo and the Tier 1 Canada Research Chair in Quantum Neurobiology. Dr. Craddock's background is in physics and biophysics, and his research brings together computational biophysics, molecular neuroscience, and quantum biology. His work explores whether quantum-level processes may influence biological systems, particularly processes connected to neuroinflammation and neurological disease. We're excited to hear about his work today, and the presentation will be followed by a community Q&A. So if you guys have questions while he's presenting, feel free to save them up and then ask them at the end. Dr. Craddock, did I miss anything? Otherwise, I'll hand stuff off to you.
Perfect. Thank you, Kira. Let's see if I can share screens here. 1 second. Can everyone see that okay?
I'm not seeing anything. I don't know if folks as well.
Hang on. Try. It is. Try this.
There we go.
Okay. And how's that? Can everyone see that okay?
Perfect.
Perfect. All right. First off, I'd like to thank the Quantum Biology DAO for inviting me. I've had some conversations with some people from the DAO over the years, and it's nice to finally connect on a formal level. So thank you for inviting me. For those of you who don't know who I am, just to give you a bit of a background, a while ago, I was an undergraduate student at the University of Guelph in Ontario, Canada, where I did a bachelor's in physics. And then I moved on to do a master's and a PhD in physics at the University of Alberta. And this is where I started focusing primarily on physics applied to biological polymers. So looking at microtubule proteins and looking at the biophysics of how microtubules work in relation to things like Alzheimer's disease, as well as the function of anesthesia. Then I moved over into the field of systems biology. I moved into the Department of Medicine, where I was building large models of the neuroinflammatory system in the brain. So looking at how neurons communicate with the hormonal system, as well as the immune system, and glia and so forth. And we're using that to predict failure modes in complex chronic illnesses, such as chronic fatigue syndrome, myalgic encephalomyelitis, and Gulf War illness. And then later on down the road, this was very similar to what we all know as now as long COVID. I then leveraged that into a position down at a university in South Florida called Nova Southeastern University, where I was recruited for a research position in the Department of Neuroscience. And I was appointed as originally as associate director and then finally as director of computational systems biology at their Institute for Neuroimmune Medicine. And so we took this idea of modeling the neuroinflammatory cascade and used it to predict multi-intervention treatment courses for complex chronic neuroinflammation. And so we leveraged that into a patent-pending process, as well as into a couple of phase 2 clinical trials for a Gulf War illness. And that work is still going on down there. But my love has always kind of been in this area of looking at quantum effects in biology. So just prior to 2024, I was offered the chance to move back up to Canada to the University of Waterloo and kind of marry this idea of quantum effects in proteins, but using it to try and find treatments or diagnostics for neuroinflammatory illnesses. And so that's what I started up as that kind of the area of quantum neurobiology and the quantum neurobiology lab here at University. So neuroinflammation is an inflammatory response in the brain. We are all familiar with inflammation. If you've ever banged your arm and you've got a bruise, we know what that feels like. It's warm, it's sore, and then it clears away after a few days. So that's an immune response to clear away debris and protect your body. The brain has the exact same thing, except because the brain is a very protected compartment in your body, it has a very specific immune response, an inflammatory response called the neuroimmune system and neuroinflammation. Now, one thing that accompanies inflammation in the brain is commonly imbalance of metal ions. So things like iron and copper, as well as magnesium and calcium. You get these changes in the homeostasis of ions in the brain, which can have effect at biological levels, as well as you get an increase in reactive oxygen and reactive nitrogen species, which is an oxidative stress response. And these, when it's short, these things can be protective, and so they protect against things like physical injury, like head trauma, mild traumatic brain injury, exposure to chemicals in low grade, or biological insults like viral exposure, something like in COVID. And so, in very short acute periods, it's protective, but when it's long lasting, these things can drive dysfunction and can lead to damage within neurons and within the brain. And one hallmark area of this is in neurodegeneration. So primarily we look at neurodegeneration as the withering of brain cells. We see this in Alzheimer's disease and Parkinson's disease and Huntington's disease. And we've kind of looked at disordered proteins with a lot of these, like alpha-synuclein or beta amyloid plaques. But more and more, we begin to believe that there's a chronic low-grade inflammation that's beginning before this, that's starting to drive those processes. So one of the things I'm particularly interested in, because I've had this work in the area of cytoskeleton, is looking at how this neuroinflammatory process leads to destruction of the protein structures in your brain, primarily the microtubule cytoskeleton, but also the actin cytoskeleton and other polymers. And the idea then is to look at the very small level scale, like how do these ions and how do these reactive oxygen species, which are molecular and atomic level events, how do they interact with proteins, and look at them at the quantum scale to try and figure out, are there ways that we can prevent this by taking advantage of maybe something we would see in that quantum level event to either predict it or detect it or to prevent it as a therapeutic. And primarily the two modes that we kind of look at to try and manipulate this is light-based or magnetic field-based. And today's talk, I'm only going to talk about the stuff that we do at the magnetic field level. I'm going to lay kind of a background for magnetic field effects in biology, as well as isotope effects in biology, and then talk about some of the work that we've been doing here in my lab. Although this is only one aspect of the work that I'm doing. So in biology, there is most biological processes are primarily explained by a standard biochemical thermodynamic principle description. However, there are observations that weak magnetic fields can measurably alter biological function. And when I say weak magnetic fields, I'm talking on the order of a few millitesla and below. And from a classical physics standpoint, or even from a classical chemistry standpoint, this shouldn't be the case, because the energies that are involved with these weak magnetic fields are below the energy that's being supplied by the thermal background. So molecules are all bouncing around due to thermal energy, and the energy of those bouncing around molecules is higher than the forces that would be generated by these weak magnetic fields through something like the Lorentz force. However, we still see these weak magnetic field sensitivities being reported. And we see things like in electron transfer, in cryptochrome and avian magnetosensing as one particular area. And so this is the European robin, and there's been a lot of work on trying to figure out how do migrating birds use Earth's magnetic field because it's so weak. How do they detect it and use it for migration and navigation purposes? And the leading candidate seems to be this radical pair mechanism. So in a radical pair, it starts off, can you guys see my cursor? Okay, you start off with a chemical reaction. In this case, it's a photo-induced chemical reaction for these birds. But any chemical reaction where it results in what's known as a radical pair. So a radical pair is when you have one electron moves from one chemical species over to another, and you're left with two unpaired electrons. And if those electrons are in parallel orientation, then you have what's called a singlet radical pair. And if they are in parallel orientation, you can have parallel orientation in one of three ways. That's why it's called a triplet radical pair. Now, the singlet radical pair very easily recombines to give you your chemical end product, whereas the triplet radical pair, the recombination of those two electrons with parallel spin is forbidden. And so that process takes either a lot longer or not at all, or it can drive it to form other products that weren't the standard chemical endpoint of that chemical reaction. Now, things like external magnetic fields or weak magnetic fields can alter whether this system ends up in a singlet radical pair system or a triplet radical pair. Now, this kind of sets up as a superposition, and it oscillates back and forth, and you can drive it to prefer one over the other by these external influences. And so that is what is believed that causes the change in rate of the formation of this chemical end product, and that is what is believed to drive a cascade that the bird can actually pick up and sense. And so this is the grand scheme of how it works, is that in the eye of the bird at the back, you have the retina. And in the retina, you have these layers of rods and cones. And in the rods, you have these segmented discs. And in between the segmented discs, you have this molecule called cryptochrome. And cryptochrome has a molecule in it that is light sensitive to blue light called flavin. I mean, it's a broad blue light. But it's also nearby some tryptophan molecules. And the idea is that when light strikes the flavin molecule and excites it, the excitation of flavin allows it to borrow an electron from nearby tryptophan molecules and set up a radical pair mechanism. And then this goes through a chemical cascade until it finally reaches down to the end excitation. And it's that completion of that chemical event that causes that cryptochrome to function, altering rhodopsin and altering the sensitivity of that rod cell, which then eventually sends a signal up to the brain and to be interpreted as vision in the bird's. And that is the idea behind the aviomagnetic compass. And so this has been studied and it's been shown. There's a lot of supporting evidence for this. There's still a lot of outstanding questions, but it seems to be a very, very good candidate for birds' ability to detect the weak magnetic field. But this isn't the only area we see it in biology. We also see it in things like circadian timing. So circadian timing is the rhythms of our body and how it aligns with the natural day-night cycle of our environment. And this also is mediated by that cryptochrome molecule. So it's not the same cryptochrome that is in a bird's eye, but it is a related molecule. Cryptochromes themselves are evolutionary conserved proteins that are part of a larger cryptochrome photolase family. And so there's all these different molecules that are related and have different functions. And so we have animal cryptochromes here and bird cryptochromes maybe even on one site, and the cryptochromes that are in our body might be over here on another end of the tree. So they're related but not the exact same. But they are all sensitive to blue light and UVA, so that they are photoreceptors for these wavelengths of light. And they usually involve a flavoprotein. And although that's been questioned if they are all technically flavoproteins, but the ones that are these ones that are photoreceptors. And these cryptochromes themselves are key regulators in biological cascades of these circadian rhythms. And so the effect of weak magnetic fields on circadian rhythms is proposed to be mediated by similar cryptochromes, but in the context of circadian signaling. So we see these cryptochromes involved in circadian rhythm in insects, vertebrates, and plants. And Cry4 is the one that is particularly involved in avian magnetoreception, but the mammalian Cry1 and Cry2 and the Drosophila, the fruit fly, are the ones that regulate that circadian clock. And so this suggests that a magnetic mechanism may affect these circadian rhythms. And so people have studied this, and they've looked at what happens if we knock out these cry proteins and see what happens with the circadian rhythm. And so we look at the period of the circadian rhythm, the natural oscillations in the body under various field strengths, and we can see that in this case, whatever this reference is for that you can get at the end, is that you can get an increase in the period with increasing magnetic field of microtesla strength. But if you do it in the knockout version, you lose that effect. And so you don't see that effect when you get rid of cry. So this is supporting evidence that potentially there's a magnetic field sensing element of cryptochromes in circadian clock. It's also been looked at in other systems in both insects and humans, and again, with supporting evidence. But other studies counteract this. So other studies have tried to produce the same effects, and depending on their experimental setup and the design that they've set up, they do not see these weak magnetic field effects. And so this is kind of a hallmark of this area of looking for weak magnetic field effects in biology, is that it's very, very dependent on your experimental setup and the way that you designed it. And so we need to be very careful when we compare two different studies, and we need to really go through the fine details when trying to say one study rules something out and another one supports it, even though they're like looking at apples and oranges. We need to make sure we're looking at the same thing here. So when you're going through this literature, read it with a critical mind. Beyond circadian timing, we also see it in things like stem cell responses. Weak magnetic fields have been observed when you apply to cell culture. We've seen it in transcription factors in leukemia cells, kinase activity in lymphoid cells, calcium levels in satellite cells, as well as reactive oxygen species in amniotic cells. You can find all these in up here in this reference 8. And it's been suggested that reactive oxygen species are an additional mechanism beyond cryptochrome, which may be driving these. So reactive oxygen species are molecules that by nature, they end up with a single unpaired electron. And because of that, they're highly reactive, and they go about scavenging for electrons, and they look for electrons wherever they can grab it, and they steal it from something else so that they can pair up their electron, but that can end up causing damage somewhere else in a system, like on a protein or a lipid or so forth. But because you have this unpaired electron, they become a candidate for being sensitive to weak magnetic fields if they're setting up a radical pair when they're stealing those electrons. So there's a study here. It's a little old now. It's from 2019 from the Wendy Bean lab, where they looked at magnetic field effects on ROS in planaria. So planaria are these flatworms that if you chop off their heads and tails, that they will eventually regrow. So if you chop this up into three portions, the middle portion will eventually regrow a head and regrow a tail, but the head will also regrow a body and tail, and the tail will also regrow a body and a head. But during this process, they're looking at, what is going on to drive that regeneration, and they found that is sensitive to magnetic fields. So they find field-dependent changes. So this is the size of the growing area, the blastema, on the anterior and the posterior end of that segment, and they can see that if you add different field strengths, that the size of that blastema changes. And it has this kind of oscillatory behavior when you start from 0 applied field up to about 600 microtesla, and they find that you can get, a decrease in size, but then eventually an increase in size. Now, this is a paper from 2019, but this same group, in conjunction with Christoph Simon's group at the University of Calgary, they just had a publication come out a couple weeks ago. So I'll point you in that direction, even though I don't have it here. I didn't get a chance to update my slides for this. Wendy Bean they went to look on this further, and looking at levels of ROS in this system based on the various applications of magnetic fields and environments. And they found that this change in the blastema is associated with ROS levels. So if they increase the magnetic field up here to 200 microtesla, you can see that you can drop the level of ROS that are being detected within the system. It's also been suggested that similar results showing ROS-based or magnetic field effects in neurogenesis are also mediated by these reactive oxygen species. So that leads us to the question, if we have both cryptochrome and now we're looking at reactive oxygen species, what other mechanisms could drive weak magnetic field effects beyond cryptochrome, or these radical pair mechanisms beyond cryptochrome? And so one of the areas that we're quite interested in is not just the application of magnetic fields, but is really looking at the application of nuclear spin effects. So we just looked at this diagram before, the radical pair mechanism. Now, this external influence I'd mentioned could be an externally applied magnetic field. It can also be influenced by a nearby nuclear spin. So if you have another molecular atom here that has a nuclear spin, it can influence the radical pairs in these chemicals, in these molecules. So nuclear spin itself is a property, it's a quantum property of the nucleus of an atom, and it's analogous to an angular momentum of the nucleus itself, and it's responsive to magnetic field. So we can imagine that we have a nuclear spin here, and you put it in a magnetic field. Is that this nuclear spin will precess around an axis of the magnetic field. Now, the nuclear spin itself is determined by the number of protons and neutrons. If you have a standard element and then you add in additional neutrons to get an isotope of that element, you can affect the nuclear spin of the atom itself. And so isotopes of the same element can have different nuclear spin. So from a chemistry standpoint, these things are the exact same elements. They have all the same electrons, they have all the same elemental properties. The only difference is that they have a nuclear spin, whereas their other isotope may not. And so classically, again, coming down from classical chemistry or classical biochemistry, this isotope change should not have any bearing on the biochemistry that's involved, because chemistry is all about electrons and electrons interacting. And so the effect of the nuclear spin on those electrons is believed to be quite weak. However, there is evidence for isotope effects in biology. So most notably, one that has been put forward in the last 10 years or so is lithium. So there's been effects of lithium isotopes on behavior and neuron function. So this is a well-famous study now made famous by Matthew Fisher and his work looking at lithium effects. It's different rearing behaviors in rats fed diets or supplements of lithium-6 versus lithium-7. And so here we have a control, and these postpartum rat mothers have average behaviors of taking care of their young. Lithium natural, which is, I think, primarily lithium. I'm going to get this wrong because I always get it wrong, but I think it's primarily lithium-7. Don't quote me on that, but I think that might be what it is. We kind of get this infrequent behavior. And so we see the same thing when we have pure lithium-7. But then when we switch over to lithium-6, we get almost the exact opposite. So as opposed to nest building being absent, we get excessive nest building. And when we have nursing being infrequent, we have very frequent. Instead of grooming the pups being infrequent, we get excessive. And it's almost a dichotomy between this lithium-6, lithium-7. So there seems to be some sort of isotope effect going on at the level of behavior in biology at this point. Other work has looked at rat cerebral cortex sustaining. So they've looked at levels of rats when they've been intaking lithium, and it's found that the cerebral cortex prefers to hang on to lithium-6 concentration as opposed to lithium-7. And so this is a study here that has looked at the amount of, lithium-6 here. I don't remember how to read this table. Here's lithium-7, I believe. I'll leave it to you. But the take-home message here is that the amount that the brain seems to prefer, at least in a rat, seems to prefer one lithium isotope over another, and on the isotope itself. Looking at hyperactivity in rats, it's also been shown that lithium-6 produce longer suppression of ketamine-induced hyperactivity than lithium-7, so another behavioral result that's an isotope effect, as well as looking at different electrical activity in the hippocampus of rats, depending on whether you're using lithium-6 or lithium-7. That being said, we see these effects at the level of behavior and cellular activity, but trying to find out what the molecular mechanism is we don't know what it is. Every time someone has looked at what is the molecular mechanism, does lithium have an effect on kinase activity? No. Does it have an effect on a certain cell type, cell viability? No. Does it have an effect on mitochondrial calcium exchange? No. And we don't know what the potential molecular mechanisms are that are driving these observed behavioral effects at the level of cells and animals. So the molecular mechanisms still need to be determined. So this is kind of an active area of investigation. I have collaborators here at University of Waterloo, Zoya Leonenko's lab and Mike Beasley's lab. They are looking very much into this lithium isotope effect. Beyond lithium, one that I'm particularly interested in is xenon anesthesia. So xenon is a noble gas with 7 stable isotopes. Let's see here, we've got, most of them have nuclear spin of 0. However, xenon 129 and xenon 131 have spin of nuclear spin of 1/2 and 3/2 respectively. Again, these are all stable. They're not radioactive. They don't decay. They're stable. They just have a different number of neutrons inside, so it changes in weight. But it's known that xenon can be used as an inhalational anesthetic. The mechanisms of how this work, nobody really knows, because xenon is an inert gas. But regardless, if you inhale it at the proper concentrations, you will undergo anesthesia, general anesthesia. And so there's an experiment that was done a few years ago now, looking at the loss of writing reflex in animals being exposed to xenon gas. And at the point where you get to 50% loss of writing reflex, it means the animal can't stand up. It's laying on its back, and 50% of the animals are falling over and do not get up. That's usually the clinical dose of anesthesia. And so here's the concentration of xenon gas to get to 50% and higher of animals losing their writing reflex. And we can see a clear difference between the different xenon isotopes here, both here in the bar graphs as well as the curves. And so there seems to be an effect, an isotope effect, that is not explainable just by mass alone. Okay, it only seems to correspond to whether it has a nuclear spin or it has no nuclear spin, not to be due to the differences in weight. But this study has not been replicated to date. I've been looking for people to do it. We've been trying to look at xenon at the molecular scale, but because it's a gas, it's a little bit difficult to work with, and we're kind of working towards that. So then I'll move on to the area where we particularly look in, which is magnesium. So magnesium is a nice isotope to work with because it has 3 stable isotopes. 24 and 26, magnesium have 0 nuclear spin, whereas 25 has a nuclear spin of 5/2. And this is very good from an experimental standpoint, because if you can observe an effect with magnesium 25, but no effect with magnesium 24 or 26, you rule out any sort of mass arguments. That way, it cannot be due to it either being lighter or heavier than the other isotope. It has to be due to the only thing that's different, which would be the nuclear spin in this case. Now, magnesium itself, my apologies, has many uses in the body. It's used in DNA replication. It's used in cellular energy, in the cleavage and synthesis of ATP and GTP. It's used in NMDA receptors and calcium signaling. It's used in bone and osteoblast proliferation. So a while ago, there's quite a body of literature from a Russian group led by Buchichenko, where they've looked repeatedly at a magnetic field effect and an isotope effect of magnesium on ATP synthesis. So in ATP synthesis, magnesium acts as a coordinating ion between the 2 phosphates at the end of the ATP or the ADP, and coordinates those phosphates so that either a phosphate can be added to ADP or a phosphate can be removed from ATP to generate energy. And so they have evidence, or they've reported evidence, that phosphokinases that are responsible for synthesizing ATP they increase with the amount of ATP that is generated increases with increasing percentage of magnesium 25 in their sample. However, these experiments have been reported to have interpretation problems and reproducibility problems. There's a famous experiment done by Craddock where they failed to reproduce this effect here with increasing magnesium 25, leading to increased ATP synthesis. However, they did find that in the presence of magnesium 25 and fields, that they did get a reduction in ATP synthesis quite consistently. That being said, I want to come back to the idea that the methods that were used by Craddock were looking at, they used a luciferase mechanism to measure ATP synthesis, whereas the Buchichenko work was looking at using a radioactive phosphate P32 label. And in that case, because we're dealing with these kind of spin effects, there may be something that is going on with the decay of that phosphorus due to the presence of magnesium spin. I don't know what it could be, but there's one potential way that something could look in there, maybe potentially explain what's going on and the difference between these two works. That being said, this is where our kind of work comes in. Because we're interested in the microtubule cytoskeleton, the microtubule cytoskeleton is made up of this protein called tubulin, and tubulin itself is a self-assembling molecule that'll stick to the end of a nucleated microtubule and cause the microtubule to grow. And as that microtubule grows, it adds on more and more of this tubulin. Now, tubulin itself has two sites that contain GTP. One of them is non-hydrolyzable, that GTP will never hydrolyze, whereas at the other site, it's a hydrolyzable GTP. And so this at this hydrolyzable site, when it's containing GTP, that's this GTP tubulin up here. And when it joins onto the end of the microtubule, after a period of time, the magnesium coordinates a hydrophilic attack on the bond between the last two phosphates and causes it to hydrolyze, removing one of the phosphates and causing a release of energy, which eventually causes a conformational change in the tubulin. And the tubulin itself bends, and it causes strain on the microtubule itself. Now, as long as there's a tubulin on the end of the microtubule that has GTP still in it, as opposed to GDP, then this microtubule will stay together. But if you remove all that GTP on the end, then the microtubule will go through catastrophe and will begin to fall apart, and it will peel back and it'll disassemble. So I like to think of that GTP cap as the plastic end on the end of your shoelace. As long as that plastic cap is there, your shoelace is intact. If you lose the GTP cap, the microtubule starts to fall apart. But if you add back in tubulin that has GTP, you can rescue it, and then the microtubule will grow. So this whole process itself is GTP dependent, but it also involves magnesium. And so because it's been shown that microtubule polymerization is affected by weak magnetic fields, we wanted to look at this further. So I was approached by a group out in Calgary, and they had mentioned that microtubules, when they're put in a hypomagnetic field environment, that the microtubules grow aberrantly. They don't polymerize as fast or as much as in the Earth's natural magnetic field. And so that didn't make sense to me at the time. I had stumbled upon this Buchichenko work, so we thought this would be ideal to try and test what's going on here. The Calgary group thought it might be a radical pair mechanism. The only place I could think of that there's a chemical reaction that goes on during microtubule polymerization is during this GTP hydrolysis. And because the magnesium is so close, we figured if we switch out the magnesium for 24, 25, or 26, we could maybe potentially see an effect. So the reason that I'm interested in this, as we mentioned before, is that microtubules are very, very important to brain cell function. So in neurons, they provide the stable structure that holds up all of the neuronal processes that are important in information processing. So they hold up the axon. They also hold up the dendrites and dendritic spines. They fill up the axonal growth cone and go out into these filopodia that are reaching out, trying to make connections with nearby neurons. And so if this system is affected by magnesium or magnetic fields, then potentially it's a mechanism that we could exploit and use to control the growth and behavior of a neuron. And that would mean we'd be able to control things like cell morphology, cell motility, or potentially things like synaptic plasticity if we could get up to the level of tissue and brains. So what we did is we followed a standard tubulin polymerization assay. So this is a standard kit that you can buy. You put tubulin in the proper buffers, and then you allow it to grow. And over a period of time, the microtubules will self-assemble and grow. And you measure that using a thing called optical density. So you pass a laser light through it, and tubulin itself is too small to scatter that light. But when the microtubules form, they're large enough that they will scatter the light. And so because of that, as the microtubules polymerize, you will get less signal passing through, and you'll get a higher optical density in the sample. And so if you measure optical density, it's an indirect measure of microtubule polymerization. So we then created our own in-house buffers where we used natural magnesium. Natural magnesium is a composition of 80% magnesium 24, which has 0 nuclear spin, 10% magnesium 25, and 10% magnesium 26. So primarily it's 90% 0 nuclear spin and about 10% 5-half nuclear spin. We then used, so we had a natural magnesium buffer, a magnesium 25 buffer, which was about greater than 99.7% pure magnesium 25, and then another buffer which was greater than 99.7 magnesium 26. And we created all the buffers identically. We did it in a big batch and then separated them out and put in the individual magnesium components. So we made sure they all had the exact same pH, and they had all the exact same concentrations of all the buffers and so forth. We then performed the tubulin polymerization assay in the absence and presence of a weak magnetic field. So we put the plates on a stand in between Helmholtz coils and put this whole setup inside of an incubator and let the system incubate and polymerize over 50 minutes, and then measured the change from prior to polymerization to post-polymerization. So we accounted for field strength, stability, temperature, pH. These are all things that could potentially affect polymerization. And then we measured the optical density, and this is what we found. So we normalized everything to the magnesium 26 on each plate with the field off condition, because the magnesium 26, we expect, it's assumed that it would have no magnetic field effect. What we found is that there was no change associated with the magnetic field or the isotope, except for the condition where we had magnesium 25 and the applied 3 millitesla magnetic field, where we had a large increase in the total amount of tubulin polymerized in this case. We're still trying to determine if this is tubulin aggregation or if this is tubulin polymerization into microtubules. So that is something that is currently still ongoing in the lab. But this is a significant effect. We've reproduced it in my lab since this was done when I was prior to moving out to Waterloo. But we've reproduced it in the lab since then. I'm just not showing it because it's preliminary work for another paper that's going out. I just don't want to put it out in a public forum at this point. But we found no significant differences between any of the other conditions. So it seems to be both a field-dependent and an isotope-dependent effect, which, from the standpoint of radical pair, it screams radical pair. So our collaborators, Hadi Zedhagigi, who's the lead author on the paper, and Christoph Simon out at the University of Calgary, they constructed a radical pair model. So first they constructed a model of tubulin polymerization, and they made this kinetic model. They made the depolymerization rate of the microtubule dependent on the magnesium state. So such that in the radical pair, the triplet state of the radical pair modifies the dissociation rate of tubulin off of the microtubule. They then constructed a Liouvillean superoperator form of the radical pair model, and then used that to predict triplet yield and the polymerization, total polymerization of tubulin over time in the presence and absence of different isotopes of magnesium and over different magnetic fields. And these are the curves that they got. And so we did not know what the radical pair would be in this system. So we took an agnostic approach. The work by Buchachenko suggests that the magnesium itself is a radical, whereas talking to a lot of chemists in the field of radical spin dynamics. They suggest that magnesium forming a radical, while it's possible, is highly unlikely for it to occur in a biological setting. You need to have some very unique chemistry going on, which probably won't be available to a biological system. So they then simulated this, and these are the results. So this, when they simulated it, they optimized it such that it would match our experimental results. And they found a unique set of parameters that does match our results. And it's within line of coupling parameters and relaxation rates and so forth that are reasonable in this kind of system. So one thing we see here is that we get both an effect due to magnetic field, either dealing with the magnesium 25 natural or 26, is you get this spike up between 0 and 5 millitesla. However, it's much more pronounced when you introduce the magnesium 25. But when you get up to very, very high levels, beyond 10 millitesla or so, the natural or the spin 0 magnesium 26 has absolutely no effect. It's very close to the amount. Now that normalized amount is set to the Earth's magnetic field here at 0.05 millitesla. Another thing that we notice here is that if you drop below 0.5 millitesla to that hypomagnetic field environment, this drops to about 60% the polymerization. So it does seem to align with that previous work showing aberrant microtubule formation in hypomagnetic field environments. But that can be rescued or improved by adding the presence of magnesium 25. So these seem to be very interesting results. We don't have the experimental points along this Mary curve, but that is what we're currently working on here in the lab. Earlier this year, a couple months ago, I received funding from the New Frontiers and Research Fund from the Canadian government to do these experiments over, but under varying field strengths to see if it does exactly align, not exactly align, but if it does show these hallmarks of a radical pair mechanism. And so that work is still currently ongoing. So in summary, the radical pair mechanism is a leading candidate to explain weak magnetic fields in biology. It's established or fairly established in flavin, in cryptochrome for avian magnetoreception. There's evidence for weak magnetic fields mediated by cryptochrome and ROS as well. There's also evidence for nuclear isotope effects. But it seems to be that a combined weak field and nuclear isotope effect can allow for added control in systems like microtubules. So I would suggest if anyone out there is looking at lithium effects, that you may also want to consider doing those lithium studies in the presence or absence of a magnetic field to see if you get any sort of change, which may hint at a radical pair-based mechanism. And so where do we go from here? Where's my lab going? We're trying to tease apart these mechanisms and try to see if we can use them for targetable purposes. We want to make sure that we're using very rigorous experiments with defined protocols that account for things like field artifacts, temperature variations, isotope impurities. And when comparing to other studies, making sure we're comparing comparable experiments, right? It's one thing to say, here I found something, and another person to come along and say, here, I just proved them that they're wrong and there's no effect. But if you're not doing similar experiments, it's in different contexts, right? There may be something else in the experimental design that's either increasing or decreasing the likelihood of seeing these effects. Another thing I wanted to point out, just let me quickly go back, is one thing that we're also interested in is due to the fact that we have this hypomagnetic field effect. We've been talking to people in that deal with space travel or extraplanet travel. It seems to suggest that if this kind of mechanism, especially in eukaryotes, is sensitive to a weak magnetic field, you may get aberrant effects the farther away you get from Earth's protective magnetic field, because these systems seem to be optimized to work around that field strength. So one, you'll get an effect if you apply a field, but also if you remove it. So it's something that policymakers or people thinking about space travel need to take into account. I think this crowd is probably aware of that. I just want to say it because I think it's important. So that being said, the references I've cited throughout the paper or through the presentation are found here. I'd like to acknowledge my team. We're rapidly growing. I have senior postdocs here that are working both at the biochemical level as well as at the cellular level. They're both looking at magnetic field and isotope effects, but we're also looking at, as I'd mentioned, light-based effects. So looking at how light is absorbed by the cytoskeleton, how that light is transferred, and how we may manipulate it by the addition of chemical cofactors. I'd also like to thank the funding agencies and my current collaborators mostly from the North American region. And I'll take questions. Thank you.
Thank you so much for presenting that all, Travis. That was super interesting. So we have a few questions in the chat, but I wanted to open it up for if anybody had questions to ask. Jeff, I saw you came on camera. Do you have a question? Would you like to start it off?
I'm thinking about it. Maybe start with ones in the chat or other people. I'm still thinking.
Sounds good. Okay, Travis, I don't know if you can see the chat, but I can read out the ones. I think this was from earlier when you were talking about the weak magnetic field effects. The question was, does the magnetic field have different effects on different species? Example, different birds? If so, why? Is there any relationship between the degree of effects and the characteristics of the species?
The short answer, I'm not 100% sure. I know that this primarily individuals have looked at the European robin. I think they've looked at other avian species. Primarily, the system that they're looking at is based on cryptochrome 4. So if it's a migratory bird and it has cryptochrome 4, then likely this mechanism could exist within that animal. I'm assuming that there would be differences in species. I can't say what they are specifically, though. My apologies.
No, you're good. And then the other one was, what role could chirality, including chiral proteins, environments play in lithium/xenon/magnesium spin effects on molecular processes of the cell and behavior?
Okay, so I know of so for chirality, I'm familiar with the chiral-induced spin selectivity, and so that mechanism is if you have some sort of chiral molecule and it's conducting electrons. Due to the chirality of the system and the physics that are involved, it can prefer to transfer a certain spin orientation of an electron such that the electrons coming at the far end of this molecule would be highly populous with one electron spin and lowly populous with the other electron spin. That being said, these are nuclear spins, and I'm not sure if anyone has looked at chirality in terms of nuclear spin. I don't know myself. There may be an effect between the nuclear spin and the electron spin in this case. And in that case, if there's some sort of dual effect there, if you have like a molecule that is doing chiral-induced spin selectivity, but you're then adding in these nuclear spins, it may play a role, but I don't know the full details of that. And my apologies again.
No, all good. And then also, people, feel free to raise your hands if you would like to ask your question verbally versus in chat. The most recent one from Guido, he asked, what role do you think the locus coeruleus might play in this context, given the neuromelanin-rich neurons may also be sensitive to red and near infrared light?
Yeah, so I'm not sure about this context. I'm not sure about radical pairs in melanin. I haven't looked at it myself. I'm more familiar with melanin in terms of photoactivation, and so melanin is an aromatic molecule very similar to tryptophan, and so you can get very interesting photophysical effects there. I'm not sure about the red and near infrared light. I think melanin has a strong absorption band down in the far blue in UV. So again, I'm sorry, but I can't comment about this one. I'm not sure. I'd also have to look into whether melanin can form a radical. So, if it's excited, could it potentially generate a radical? I'm not sure on that as well, but it's something that someone could look into and see. It might be able to explain that.
Yeah, totally. All right, are there any other questions from folks? Like I said, feel free to come on camera, should it please you. Ash, you want to go? Or Jeff?
Oh,
Hi,
Ash, go ahead.
Travis. Yeah, thanks for a great talk. It seems to have got dark since the talk started here, and I think I can't tell if my camera's cut out entirely there. I'll try and put it back on. Anyway, yeah, so having worked in startups with self-assembling peptides, I've found your work inspiring over the years. It was great reading about the superradiance in microtubules in tryptophan, and I've always been interested to find out if there's any more developments about looking at coherence and how far coherence could spread through that mechanism. And it would be interesting to know if any work's been done looking at that while varying the magnetic field background.
Yes.
I think Christoph Koch was maybe doing some work looking at xenon effects. I don't know if there was any more that was done on that recently.
Yeah, I am aware that Christoph Koch is trying to look at that xenon. It's interested to see what comes out. In regards to the energy transfer in tryptophan aromatic lattices, there has been work. We have a couple preprints that are up, as well as a couple recent publications based on that, as well as and a review article that just came out recently as well in Journal of Physics Photonics. So there's the work that was done by Babcock and Kurian, where they looked at measuring the quantum yield of tryptophan, or sorry, of tubulin compared to microtubules, and they found with increasing, from modeling standpoint, with increasing size of the microtubule, you get increasing quantum yield. And then they have experimental evidence that shows from microtubules compared to tubulin that there's a fairly large increase in the quantum yield. We've reiterated that experiment and found very similar results. However, in our modeling, we did a closer to biology kind of based approach. So in the original model by Babcock, it's a highly idealized structure. It's just the same protein structure repeated over and over again with no variation. We added in molecular dynamics, and so that the tryptophan orientations and such are more closer to a biological system. And we found that effect is the increase is still predicted, but it's much less than is predicted by their modeling. We still do get an effect that we see experimentally. And then we showed that you can manipulate it by the addition of additional chromophores. So we added in additional tryptophan molecules to the system and found that it modified the quantum yield in a way consistent with the superradiance model. That doesn't mean that it is a superradiant effect. It just means that it is consistent. I don't think that the quantum yield measurements themselves are a smoking gun of a quantum biological effect. They are consistent with it, but I think you would need to do more in-depth photon statistics and ultra-fast measurements to try and determine whether it's truly a superradiant quantum effect or not. Along those lines as well, we have some new recent work showing that tryptamine molecules, so tryptamine are indole-based molecules, they bind and will interact directly with the microtubule cytoskeleton. So we have experimental work that's in review right now. I believe it's out at a preprint. We've done modeling of where they bind, and we have experimental evidence showing they do bind, and they do, in some cases, promote microtubule stabilization. And it's predicted that these will also alter the superradiant properties, and so that's something we're currently looking at is doing these same experiments over again by adding in these different chromophores and these different light sensitive molecules on the surface of the microtubule. So stay tuned. There's more coming out.
That's really interesting with the link between the indoles and psychedelics as well. So thanks. That's fantastic, and I'll have a look at some of those preprints.
Okay, excellent. Yeah, if you can't find them, just send me an email and I'll get them to you.
Thank you.
Go ahead, Jeff.
Hi, Travis. Thanks for the presentation. Really enjoyed it. It strikes me that isotopes is a way of isolating spin effects from regular chemical reaction effects. This is really interesting, sort of promising route. Is it possible to create, and you may have said this, and I'm learning this area, so forgive me, but is it possible to create isotopically distinct versions of tubulin and see whether they just behave differently under the geomagnetic field. It's like if you imagine the geomagnetic field is doing something to tubulin, and
Mhm.
that's happening by means of spin, then if you put in isotopes with different spin properties, maybe the tubulins don't form the microtubules in the right way. Do you see what I'm
Right. Yeah. No,
pointing
I
at?
get what you're getting at. And I mean, and there are, you can add, you'd have to synthesize it, it'd be a process, but, you supply a different version of carbon or something like that when it's being created. Um, then you could have different spins located within the tubulin protein. Um, but you have to be careful when doing this because that's why I like working with magnesium, because you have that light version, no spin, heavy version, no spin, and then that middle version that does have a spin.
Right, okay.
And certain molecules, you don't have that option, right? So I think phosphorus, for example, I don't think there is a version. I think they're all spin one. Could be wrong on that. Could be wrong on that. Or it only has maybe two. For example, lithium, as an example, only has two stable isotopes, and so we wouldn't be able to ever rule out whether it was due to mass or not. But molecules that I know that are biologically relevant that do have this ability are calcium,
Hmm.
zinc, and magnesium. Um, Magnesium is by far the more abundant one that primarily it deals with. When I say abundant, there, when you get into
Hmm.
like say calcium or zinc, it's like 1% or less. So
Hmm.
naturally, it's not available, and
Hmm.
so to try to extract that out and get the purified version of it for experiments can be quite costly. Magnesium itself, having that for 10% magnesium 25 naturally available, um, it means that it's always there. So that itself might be mediating some magnetic field effects that we're seeing. But in addition, like it was shown in the rat cerebral cortex where it prefers lithium six, there's other evidence of other biological systems preferring a particular fraction of isotopes. Not necessarily based on nuclear spin, but it's just that the biology they prefer a given isotope over another isotope. And if that's something that's going on in our biology, let's say our biology prefers magnesium 25 and it's selecting it for some
Hmm.
reason, then being sensitive to magnetic fields probably increases with that selection process. Or it could be selecting to not have it. Maybe we don't want to be sensitive to magnetic fields. It's like, no, keep this stuff out of us. And it would allow us to supplement in some way to try and increase those values if we want to see what goes on. Anyway, that's
Thanks, appreciate it.
no worries.
Yeah. Josh, would you like to ask a question?
Yeah, yeah. Travis, good to see you again. It's been a minute since we talked, but I thought because when you mentioned selection, I mean, immediately this is very interesting to me because, like, I mean, are you familiar with like Michael Levin's work with bioelectric like reprogramming in kind of this in planarian as well? So I think that's very interesting in connection to the work that you cited with Van Heusen's work on the weak magnetic fields, because then it seems like you're basically changing what would be, I guess, the configuration space. I mean, from quantum field theory's perspective on the electrons and whatnot in atoms or in molecules, and magnesium would all be essentially manipulating the environment that is, you know, manifesting selection in some sense. So I guess I'd be interested to hear your sense would be what's being manipulated here to then change what's being selected as kind of what you were discussing in that regard. So any thoughts that come up to mind on maybe the intersection between those different ways to manipulate the configuration space potentially?
What is the mechanism by which biology would select a given isotope
Yeah.
form? I mean, yeah, I mean, if it does select a given isotope over another one, then there would have to be some sort of mechanism that would be um, sensing not just the electron configuration, but that the way it's coupled to the nucleus, right? Because that's really how it's mediated. It's, spin orbit coupling type of interaction that's going on. I don't know the full details of how that is for someone smarter. It looks in this area, but I think the first round that we would want to look at is doing the experiments and looking at take biology and just measure the abundance of given isotopes that might be of interest. So when you take like a system and you look at, okay, what is the calcium breakdown here in terms of isotopes, and if you find something that is different than what is available naturally, like what you get out of the ground, then that's pretty strong evidence that system at least is selecting for a given isotope over another isotope.
I see, yeah, because
the exact same breakdown as naturally, then we're just taking in and we're keeping everything status quo.
that makes sense because Levin calls it like interfacing with the Platonic space, which I guess would be very similar to, a configuration space in quantum field theory. So then the idea would be is that the environment is what's forcing the selection. So if you change the environment, new things can be selected because you're reducing the essentially like the path integral's natural flow of energy or some sort of something like that. So that's very interesting. Yeah. So thank you.
Yeah. All right. Thanks, Josh.
Travis, I know we're hit one. If you need to run, let me know. I want to be cautious of your time.
Oh, that's okay. I see a couple other questions here.
Yeah, do you want to? I think there's.
Yes. It looks like Jeff's running, but I'll see if I can answer it quickly. One other question: the DAO has access to a hypermagnetic chamber, less than 1 nanotesla. Are there any microtubule studies we could run that would be helpful? Yeah, I mean, reproducing just a polymerization assay of microtubules in that hypomagnetic field environment. I mean, when I'm pointing to this paper done by Wang, and they show these hypomagnetic field effects, that was one experiment that was done quite some time ago. And I believe they use a different protocol than what is the standard protocol for something like cytoskeleton ink, which is where we get most of our tubulin source from. It's a very standard assay. And so if you just did that assay in the hypomagnetic field environment and confirmed, is there a difference, yes or no, right? I know that we all want to do new studies, but reproducing some of the old stuff and then expanding on it once you see the results, I think is also very important. One thing, if you are thinking about doing that, is being very careful that it's got to still be an incubated environment, right? Because it has to maintain a constant temperature. So I think probably if you have a hypomagnetic chamber, I'm assuming it's in an incubator, but if it's not, that'd be something you have to consider.
Yeah, totally.
Then,
I think there's two from Prama.Institute, one above Jeff's comment, one after. Is there any modulatory effects known of nuclear spin on voltage-gated calcium channels within magnetic field?
I don't know about that. I'm not aware of that. I don't know, but I know that people have asked me. I was just at a recent meeting where there's a bunch of heads from funding agencies, and they were talking about these weak magnetic field effects, and they were asking, what is the effect of weak magnetic fields on ion channels? And they wanted to say, like, does it change the flow of ions that are going through an ion channel? I don't know if there's been a lot of work on that. If you look at the forces that are involved, like I mentioned, it's much smaller than thermal environments. So from a back of the envelope type of calculation, it shouldn't really have an effect. But again, when you actually do the measurement, if you see something, you see something.
Yeah. Then the last one, I'll count this as the last question.
Could you say that nuclear spins of various biological structures could form a type of an entangled attractor at the topological field, or I'm assuming field, responding to space-time via interaction with local magnetic field? Ooh, that's a tough one. My question would be entangled with what? Like entangled with itself or entangled with the field itself? I'm not sure what the question is with itself. Okay. I'd have to think about that one. That one's getting a little bit beyond my thought structure at this point. I mean, anything's possible, but to get into the details of it, I'd have to think it out. Um, unfortunately, not unfortunately. So, like I said, I started out as a physicist, and I was very into condensed matter physics and Hamiltonian and whatnot. And over my career, I've moved. Now in the Department of Biology. I feel a little bit far away from physics now, so I'm trying to come back to it because it's been a while since I've been there. I've also kind of stepped out into the area of experiment, and I realize where, because it's such an interdisciplinary field, being kind of now on both sides of physics and biology, I see why, when physicists explain why something is important, why biologists aren't understanding it, like why should we care about it? But at the same time, when biologists are explaining what's going on and the physicist doesn't quite understand why the experiments are the way that they are, I'm kind of seeing that as we go here. And so I think that I encourage people that are in this field to really hop the fence and get your hands dirty on that other side, because it'll open up your kind of viewpoint on everything that's going on.